Good, good. Miles, let's take a look at polling question number, well, I guess 3, as it were, our last polling question of the day. What indications are you addressing with mRNA therapeutics? Oncology, infectious disease, genetic disorders, or other? So while our audience is taking a look at that polling question, Doctors Van Cott and Atkinson, if you're looking into a crystal ball, would you add anything to that list, that multiple choice question we just asked? Is there anything or does that cover the gamut, you think, for the foreseeable future?
I think
am I asking you a speculative question that you're not allowed to answer?
Go. I'll let Tom go first, and then I'll jump in.
No, I think, I mean, obviously those are enormous buckets, right? And enormous indications. I think what you're seeing in there, though, is, for example, how we're attacking genetic disorders with mRNA, right? Or how we're attacking oncology, whether or not it's with, immunostimulants that are, directed to the tumor, whether or not it's something like an in vivo CAR T application, with genetic disorders, it's, is it in vivo administration of a CRISPR-Cas9 mRNA or ex vivo, modulation of different cell types. So I think the 3 categories are right, but they're just within each. It's extremely broad in the applications towards each.
Yeah. Those are really big buckets. I do think it's about where you can deliver it as well. I thinking of like the blood-brain barrier, right? Can we somehow, people are working on, can we somehow get across and start working with neuro diseases, right?
Yeah.
Not that those don't fall into that category, but one of those categories, but it is, it's a delivery area.
Yep. Yeah.
I mean,
Miles, don't show the results yet. Don't show the results yet. We're gonna put bets. We're gonna see who's right here. I'm sorry, Dr. Van Cott.
I was just gonna say. I mean, there's companies that are trying to develop vaccines against chronic diseases. And if those are effective, there's no reason why an mRNA approach could not improve upon that. So I think it's wide open as far as amplifying those or increasing those different ABC buckets you have there.
Yeah. Okay. So as we sit today with these large deep buckets, Dr. Van Cott, which one's going to lead?
Well, I would say, it's a very dynamic. I'd say if it was a year ago, we'd say B, but I'd say today we're probably in A in oncology.
All right. Dr. Atkinson, would you agree?
Yeah, I'd agree with that. A couple of years ago would have been B, but definitely A today.
All right, let's see where we're at, Miles. They nailed it. They nailed it. It was close. Well, it was close, but you guys nailed it. Yep. Very good. Let's talk.
That would mean that the other one, right? I mean, I can only see the first 2, but then that would have been like 30%, right? So all pretty close.
Oh yeah, I get well, genetic disorders and other, together they hit about 30%.
Yeah, they come in together at 30%.
But you were both right. Oncology, the recent trend. In the time that we have left, I've got a bunch of regulatory questions for you guys. A couple of which I think we already hit on a bit. So let's jump into some of those. What additional parameters do you anticipate regulatory agencies applying to mRNA therapeutics in comparison to mRNA vaccines? And I know, again, Jeff, like you said, a lot of this is sort of going to be case dependent. But can you make any general statements about it?
Yeah, I mean, I don't know if it's additional. I think it's just more focus on certain areas, right? Again, how much the dose is just so much larger for the therapeutics than it was for the vaccine. So there's going to be a lot of sensitivity around that. And thus that drives, we wanted more understanding. I don't know that I'd say, it's additional parameters, but more understanding around the impurities and the mRNA integrity. I think there are a lot of things out there that they'll want as characterization, but are not really practical yet from a release standpoint. So So I think that they'll be asking more and more about the impurities, but they'll ask some other things like probably, what about, empty nanoparticles? Does that matter? What about morphology? What do they look like? Do they all look the same? What's your distribution of different types? So I think those are things that we're already looking at. But I think there'll be an expectation that that's in a package as part of your understanding of the product.
Yeah. Another sort of general question that just popped into my head: is it safe to assume? So, I mean, traditionally, right, historically, it's well accepted that the vaccine approval. I say historically, pre-COVID pandemic, the vaccine approval process has been lengthier because it required longer time studies, obviously, and obviously because we were putting a therapeutic or a vaccine into a healthy population. So generally, I think it's safe to say, like, those approval processes and development processes, they took longer than maybe therapeutic. First of all, would you say that's a fair statement? No?
I might let Tom answer that. I don't have a ton of experience in vaccines, so I don't know a whole lot about the development piece of that prior to COVID. So I don't know, Tom, if you've got any additional insight into that.
Yeah. So let me just go back to the previous question too. I think what Jeff said was absolutely right. But I was just thinking when he was answering it, two other parameters I think that the FDA may want to be looking at, when we go to these larger doses. I think one is going to be biodistribution. It's interesting looking at kind of the packets for the COVID vaccines. There was very little biodistribution studies done. And again, those are much lower doses. So I think they're going to be very interested. And that gets at that very first question we talked about, is where are these LNP, where are they going? Where is it being expressed? So I think there's going to be a lot more interest in where these products are distributing throughout the body. And of course, when we have these targeted LNPs, or when we have engineering the mRNA, you'd be able to see some differences. And then second, I think also, especially when you're doing therapeutics, they're going to be looking more at immunogenicity, the immunogenicity of the LNP itself, what kind of response is being generated. Hopefully none, right? Mm-hmm. But I think they're going to be looking a little bit more, especially, the higher dose and repeat administration. So I think to Jeff's great list, I would just add biodistribution and immunogenicity as two additional parameters the FDA will want to look at for these therapeutics.
Yeah. Very good. Yeah, the question I was getting at, and I'll try to be more concise about it, is: do you anticipate that given the traditional, I guess, trajectory of vaccine development and that chronology, taking that continuum being sort of lengthy, now mRNA comes into play and we demonstrated great speed. Obviously, companies like Omega and Combined would love to play off of that speed in their own commercial efforts, commercial endeavors. Has there been a tempo shift as it relates to therapeutics or vaccines, for that matter, that are mRNA based? Has there been sort of a tempo or cadence, a measurable or palpable sense that the cadence is increasing, or not so much?
I kind of look at it in a couple different ways. One, when we talk about the rapidity, how fast the COVID vaccines, which really was incredible and remarkable. When we talk about, say Operation Warp Speed, what that really did was funded and de-risked a lot of the steps in clinical development. So instead of being sequential, they were simultaneous. Usually, you do a small batch, you do a phase 1, you don't start ramping up commercial production during your phase 1, and so with the de-risking by the government and with the funding, these companies were able to start commercial development while in phase 1 and prepare for, say, phase 3 trials. That really, really shortened the timeline. I don't really see that happening, again. for your standard development, I think your clinical development will go back into sort of a sequential time frame. But I think things like the FDA accelerated, approval rate, sort of like what they do for rare diseases, if you get into that track, phase 1, phase 3, commercial, if you're on that track, you can accelerate your clinical development. But I still think, the bulk of a lot of your time is still, and Jeff, you can obviously add even more, is in your discovery and, identifying and down selecting your top product that you want to move forward. And I don't know, I mean, mRNA can certainly accelerate your ability to make multiple products to evaluate, but that still is going to be, a long period of time to be able to optimize and down select lead candidates.
Yeah, I mean, I'll concur with that. There's a lot there that makes it maybe better than traditional drug discovery. That's already sped up, but it's figuring out what you're going after, and then figuring out how to make it get delivered to that target, to those cells, and then you've got all the stuff afterwards, which, I think there are good ways to speed up portions of that process that were kind of already underway on the accelerated approval process. Yeah, but I agree. I don't think we're going to see companies going ahead and putting all that money at risk to scale up, to move faster.
Yeah, it's I'm reminded of a conversation, Jeff, that you and I had with the guy who I presume to be your boss, Shiva Sakamori, SVP and head of tech ops over there. Is he your boss, in fact?
He is, yes, yeah.
Yeah. He in that conversation, he said speed doesn't necessarily help establish good practices, which I kind of found paradoxical coming from a company striving to become a commercial therapeutics manufacturer. So give me a little bit of insight into that balance between speed and GMP expectation. Like, I mean, I get it. We can't move too fast, but
yeah, well, I mean, there's obviously some things that you can't compromise on, right? Safety being number one, right? It's got to be safe. And so there are certain steps that are just going to take the time that they're going to take. But the way we balance it is using a risk-based approach, which has been talked about for years. And I think a lot of people have really started incorporating this, but taking the understanding you have and trying to figure out what is our risk if we don't do this particular experiment, but we move on to this one. So there are ways to manage some of that. The part that you have to be careful of is that don't make that decision and then you find out later that maybe that was a bad idea. So it's hard, and you need a lot of experience to figure out if what you're doing is a reasonable thing to do. And then the other part is, don't always, this gets a little bit away, I'm a little contradictory, I guess, from a platform approach. Don't just assume that everything is going to be exactly the same, right? Just because it was done this way at one time doesn't mean it should be done this way the next time. So it's a stop, think, evaluate, and then move forward after you've done that evaluation.
Yeah. We're at the top of the hour. It's like the fastest 60 minutes in the biopharma business, the Bioprocess Online Live, right? Like the fastest 60 minutes of your day. But I want to give you an opportunity, Dr. Van Cott, to maybe comment on Jeff's response just now, or offer us some parting shots.
No, I think, Jeff, that was a really nice summary right there. No, I think this is, obviously this is an extremely exciting field. There's been a lot of developments already, but I'm just amazed at, how much incredibly good work is being done daily, just keeping up with the innovations in the mRNA design itself, the different, non-viral vectors that are being used to deliver, the assays that are being delivered, the indications that are going after. It's really exciting to be in this field and trying to keep up with all the innovation out there. So thanks a lot for inviting me on to this webinar.
Oh, it's my pleasure. It's my pleasure. It was great to have you, Jeff. It was great to have you as well. I know we didn't get to everything that we wanted to discuss, that we originally talked about discussing, but since Dr. Van Cott already committed at the beginning of the show, he already committed to making this, a special series. We'll have you both back, and we'll have another opportunity to get into some more detail around the platforms and that kind of thing. So,
Fantastic.
yeah, but in the meantime,
Yeah.
thank you both for joining me. I thought it was fantastic. You both did a great job, and thank you to our audience. That's about all the time we have. I do want to remind you that this program will be available on demand within 24 hours of closing out today. So do share it with your colleagues. Bookmark bioprocessonline.com as a resource for peer-driven biopharma development and manufacturing content. Be sure to subscribe to the Business of Biotech podcast that I host wherever you listen to podcasts, and have a great day.